Thanks for the kind introduction, Lauren, and I am excited to share an update on what my lab has been working on trying to use chromosome engineering to understand and correct alterations affecting the P arm of chromosome 8. And as I'm going through these slides, if you have any questions or you want me to explain anything in more detail, feel free to just unmute and jump in. And I'm happy to provide any additional information or add any answers that I can to help explain things. So my lab is based at Stanford University. And in general, we are interested in using genetic approaches to study cancer and development with a particular focus on chromosome disorders. Um, and I want to start off right at the top by acknowledging the people who made the work that I'm going to share today possible and who did the actual work. So Sophia was an amazing research technician in my lab who did most of the experiments that I'm going to tell you about. I believe she's on the call today. Additionally, for some of the experiments with iPS-derived neurons that I'm going to tell you about, these were done by Stefan Pinter and Aaron Banda at the University of Connecticut. I'm also extremely grateful to Project 8P for financially supporting this work, and of course the 8P heroes and their families who donated the cells that we used in this project. I'm incredibly grateful for their assistance and their contributions to what I'm going to tell you about. So the overall goal in my lab is to use chromosome engineering to study and potentially to treat disorders caused by chromosome rearrangements like cancer and different developmental syndromes. The most common developmental syndrome associated with a chromosome disorder is the trisomy of chromosome 21, which causes Down syndrome. So this is the karyotype of a human woman. And you can see that there are 3 copies of chromosome 21 here. These are the chromosomes from a woman with Down syndrome. And our overall goal is to develop ways to study chromosome changes like the trisomy of chromosome 21 in the lab to understand how it impacts normal development, to understand how it impacts processes like cancer, and to try to develop approaches to address some of these consequences of chromosome rearrangements and chromosome copy number alterations. And initially, when I first connected with Project 8P a few years ago, one of the first things that we set out to do was to try to develop a new system in which we could use chromosome engineering to study the consequences of rearrangements affecting the P arm of chromosome 8, of rearrangements that cause 8P syndrome. And I want to take a few minutes to explain the value of this, or ideally what the value of this would be, and how I think it can benefit the goals of the foundation overall and our overall understanding of development and different developmental syndromes. So imagine for a minute that we have cells that were graciously provided by an 8P hero. So we just have cells growing in a petri dish. They could be any type of cells. They could be stem cells. They could be neurons. They could be blood cells. Well, we can take these cells and then we can do different experiments with them. We could study how these cells produce neurons and cells in the brain. We could perform a screen to try to find drugs that block seizure activity in these cells. This would be a useful research tool. However, for any type of experiment like this, where we were just studying cells from an 8P hero in isolation and doing drug screens or doing genetic experiments, there would be a very significant limitation to these experiments. And that is for anything that we discover, using these cells, it wouldn't be immediately clear whether the discovery that we made, whether the beneficial drug that we found is specifically caused by the 8p rearrangement or whether it was caused by each person's unique genetic background. Because, every 8p hero is unique, every individual on the planet is unique, and there are all sorts of just genetic changes between individuals walking down the street. And so if you're studying cells from an 8p hero in isolation, you can't necessarily know whether what you're studying is caused by the 8p rearrangement itself or just normal genetic differences between people. One way that this is sometimes solved is you can study as the parents of an affected individual. The parents of an 8p hero will donate their cells as well. And so you can compare the cells from an individual's parents with the cells of an 8p hero. But even when you're comparing parent to child, parents and children are 50% different. And so that isn't a perfect solution either. So as an alternate approach, as a way to potentially solve this problem, what we set out to do is to try to create genetically matched cells that either have or that lack the rearrangement on chromosome 8. So we can have cells from an 8p hero. And then if we could, say, wave a magic wand and then eliminate the rearranged copy of chromosome 8 from the cells donated by that hero, then these cells would be genetically identical except for the rearranged copy of chromosome 8. And then that gives us a system for studying these chromosomal rearrangements where the only difference is the presence or absence of the altered copy of chromosome 8. So if we had a matched set of cells like this, we could then study how brain cells are formed in each cell population. And if we see a difference between these two populations, we know that the only difference between them is the altered copy of chromosome 8. And so we can say that that's actually causing this difference. Or if we did a screen for drugs that block seizure activity in these cells, again, we know that the only difference between these two hypothetical cell populations is the rearranged copy of chromosome 8. And so we can know that we are directly studying the consequences of this rearranged chromosome. So this gives us a genetically matched, a well-controlled system for isolating the effects of the rearranged copy of chromosome 8 from all the other genetic differences that happen between people, that happen between parent and child. This gives us a way for really isolating what this rearranged copy of chromosome 8 is doing. I think for 8p syndrome in particular, there is also an additional very important reason why chromosome engineering to address the altered copy of chromosome 8p is very important. And that reason is that we know very well that every 8p hero is unique. So this is a published diagram of the spectrum of alterations affecting the P arm of chromosome 8, with deletions and duplications here you can see around the low complexity region on chromosome 8p. And one thing that I think we can very well appreciate is that no two 8p heroes basically are identical, that the span of the duplication and deletion are different throughout this population. And so I think what that means is that an approach looking at this syndrome from the chromosome region may be more broader and applicable to more affected individuals than a single gene approach. Because if you're studying, say, a single gene that's located close to the centromere on chromosome 8p over here on this side of the chromosome, that gene might be altered in several individuals who have 8p syndrome, but there are dozens and dozens of other individuals who where that gene isn't altered in them. The thing that unifies all these diverse heroes is the fact that chromosome 8p itself is affected. And so, if we were able to develop an approach that would, broadly speaking, restore a normal karyotype and restore a normal copy of chromosome 8 to cells that had all sorts of different arrangements on chromosome 8p, that could potentially be beneficial for and address all sorts of different rearrangements where a single gene approach might help some individuals with 8p syndrome, but because of the diversity of rearrangements that we see, it wouldn't be broadly applicable for all rearrangements. The final reason why I think chromosome engineering is important, and by chromosome engineering, I mean trying to develop approaches to correct chromosome rearrangements and restore a normal karyotype, is I think that this is potentially a, um, a possible therapeutic approach with many, many, many hurdles to surmount first. So, 8p syndrome is one type of chromosome rearrangement. There are many other uh, developmental syndromes caused by chromosome conditions as well. Turner syndrome and Klinefelter syndrome affect the sex chromosomes. Down syndrome affects chromosome 21. And there are many investigators out there, including my lab, who are starting to think about how chromosome engineering could be used as a therapeutic approach to potentially treat some of these conditions. So, for instance, there's a recent publication in the academic journal Science where these researchers were able to produce fertile offspring in mice that were affected by a sex chromosome disorder. Individuals have also worked for a number of years on trying to address Down syndrome most commonly using different chromosome engineering techniques. So there are all sorts of hurdles, in the way that make this incredibly challenging. None of this is going to happen immediately in patients. But I think that there is exciting research being done right now in cell culture systems and in mouse models that underscore how chromosome engineering may be useful as a therapeutic approach to treat diverse chromosome disorders in the future. And this is something that we're very excited about. So, with that as a background, my lab set out to use chromosome engineering to try to improve our understanding of the causes and consequences of 8p syndrome. And I'll tell you about today what we've done toward that goal. So, our initial approach was to develop a chromosome engineering process that would allow us to eliminate the altered copy of chromosome 8p from cells that were donated by an 8p hero. This would then give us this genetically matched pair of cells that I told you about, where we could know that the only difference between these cells is the rearranged copy of chromosome 8p. There are no other genetic changes in this population, and so we can say, with a high degree of confidence that any changes we see when comparing these cells is caused specifically by the alteration on chromosome 8p. And then secondly, we wanted to use these pair of cells, if we were able to successfully generate them, to characterize the effects of the 8p rearrangement, particularly looking at the effects of the 8p rearrangement on gene expression and looking at the effects of the 8p rearrangement on the production of neurons from these cells. So I'm going to start out by telling you how we were able to achieve this. So we're thankful to Project 8p for providing us with cells from an 8p hero. These were induced pluripotent stem cells from an 8p hero who has an inversion duplication deletion on chromosome 8p. So here's the duplication region in red on chromosome 8. Here's the deleted region in blue. And this is a picture of the karyotype of those individuals determined by whole genome sequencing. Basically, you can see that most chromosomes in these cells are present in 2 copies here. You can see the duplicated region in red is present in 3 copies on chromosome 8, and the deleted region is present in blue, is present in 1 copy on chromosome 8. And this is what their karyotype looks like, determined from whole genome sequencing. So we received these induced pluripotent stem cells from the Project 8P, from an 8P hero. And then our goal was to eliminate this rearranged copy of chromosome 8 in these cells and then restore a normal euploid set of chromosomes, which we could then use to do a number of experiments with, like I previously mentioned. However, Sophia, our amazing technician who was working on this project, we started out trying all sorts of different techniques to restore euploidy in these cells, and we got a lot of negative data. And I'm not going to walk through all of the negative data for the sake of time. Happy to answer any questions about it. We tried a number of different approaches to eliminate the rearranged copy of chromosome 8, but initially none of it worked. However, while we were doing chromosome engineering on these cells, basically spontaneously, we got a cell population that we thought would be useful for further experiments. And that is, while trying to target chromosome 8 for missegregation, Sophia was able to spontaneously isolate a clone of cells that had an extra copy of chromosome 8, rather than a lost copy of chromosome 8. So this is a picture of the karyotype determined from whole genome sequencing of this iPS cell population. These were the cells provided by Project 8P from an individual with an inversion duplication deletion on chromosome 8. And we isolated a clone that had an extra copy of chromosome 8. So instead of having 2 copies of 8q, 3 copies of part of 8p, and 1 copy of part of 8p, this clone had 3 copies of 8q, 4 copies of part of 8p, and 2 copies of the deleted region of 8p. And while Sophia and I were thinking about this, we realized that this might be useful from a chromosome engineering perspective, because we thought that if these cells now have 3 copies, they have an extra copy basically of a normal copy of chromosome 8, they might tolerate the loss of the rearranged copy of chromosome 8p better, because then just losing the rearranged copy by itself would restore a euploid karyotype in a single missegregation event. So we thought that this could be a useful intermediate cell population that would help us towards our goal of isolating cells with a normal karyotype. So we started working with these cells, and we applied a new approach developed by our collaborators at New York University, Teresa Davoli's lab, for targeting chromosome 8. This new approach was published in parallel to the work that we were doing. So the Davoli lab at NYU developed an approach called karyocreate, where they use CRISPR-based technology to induce chromosome-specific missegregation. In this approach, they use the Cas9 enzyme, which can target a specific DNA sequence, and they fuse the Cas9 enzyme to a mutant kinetochore protein, which causes the production of a faulty kinetochore on a chromosome of interest. And the kinetochore is the region where microtubules attach to a chromosome during mitosis Microtubules are what pull chromosomes apart during mitosis. And by creating a faulty kinetochore, you block the segregation of that chromosome. You basically prevent that chromosome from being pulled during mitosis, leading to chromosome missegregation. So basically, the DeVoley lab published an approach that allowed us to target a specific chromosome for missegregation. We collaborated with Teresa's lab to develop an approach to use this for chromosome 8. We applied it to the induced pluripotent stem cells with the trisomy of chromosome 8 that we had spontaneously isolated. And we were very excited to see that following the use of this new karyocreate technology, we were able to successfully eliminate the extra copy of chromosome 8 from these cells. So following the use of this karyocreate technology, we were able to isolate iPS cells derived from the 8p hero with an inversion duplication deletion of 8p, in which we had restored a wild type revertant karyotype, in which they had 2 normal copies of chromosome 8. So to put these side by side, we started out with cells derived from an 8p hero with an inversion duplication deletion on chromosome 8p. We went through a kind of prolonged process of chromosome engineering involving the generation of a trisomic intermediate and the use of this new CRISPR-based karyocreate technology. And then we were successfully able to isolate derivatives of the cells from the 8p hero in which a normal copy of chromosome 8 had been restored. So in these 2 cell populations, chromosomes 1, 2, 3, 4, 5, 6, 9, 10, so on are genetically identical. All of the SNPs are the same, all of the mutations are the same. The only thing that we did is we eliminated the rearranged copy of chromosome 8 and we restored a normal copy of chromosome 8 in its place. So this gives us an excellent genetically controlled system for now studying how this rearranged copy of chromosome 8 impacts different aspects of cell biology, gene expression, brain development, and other processes that we may be interested in. So we set out to use these 2 cell populations to explore the consequences of chromosome 8p rearrangements. So I told you we were able to successfully remove the rearranged copy of chromosome 8p from cells that were provided by an 8p hero. And then we set out to use this pair of cells to characterize the effects of the 8p rearrangement on gene expression and on differentiation into brain cells. So we started out by just doing RNA sequencing on the cells with the 8p rearrangement and on the cells where we had eliminated the 8p rearrangement. And we also had cells that were provided by the parents of the individual, the 8p hero as well, that we also did RNA sequencing on. So this is a bar graph showing the quantification of differentially affected genes, basically genes that were significantly overexpressed or significantly underexpressed in 3 comparisons. We have the proband, that is the cells with the inversion duplication deletion of 8p compared to cells provided by the parents. We have the proband versus the revertant. These are the cells where we engineered the elimination of the rearranged copy of chromosome 8p. And then we also compared the parents with the revertant. And then we looked for genes whose expression changed in either the deleted region on chromosome 8p, the duplicated region on chromosome 8p, and then we looked at all other chromosomes as well in these individuals. So from this analysis, I think we found a few things that really stood out and that were really interesting to us. So the first is that comparing the cells with the inversion duplication of 8p to the revertant, that is the cell population where we eliminated the rearranged copy of 8p, allowed us to significantly narrow down the number of genes that contribute to 8p pathology. So if we just looked at the cells from provided by the 8p hero and compared them to their parents, we had something like 1,400 differentially expressed genes. That is 1,400 differentially expressed genes that were upregulated or downregulated or otherwise differentially affected. However, when we looked at the cells, when we directly compared the cells with the rearranged copy of 8p to the cells where we had eliminated that rearranged copy, we only had about 350 differentially affected genes. So we were able to kind of narrow our search from 1,400 genes to 350 genes just by comparing these cells in a genetically matched context. And I think this also underscores how just doing a comparison between parent and child, you get hundreds of differentially expressed genes just based on the fact that parents and children are not genetically identical. But when we look at the effects of inversion, duplication, deletions in a genetically identical context using chromosome engineering, we can significantly narrow down the number of differentially affected genes that we're seeing, which can, I think, help us focus in on ideally uncovering the genes that are most important for understanding this condition. So that's, I think, one thing that we found from this. The second thing that we found from this is that maybe surprisingly, most genes that are affected by inversion, duplication, deletion 8p are not actually on chromosome 8p. So comparing the proband versus revertant, that is the cells with the 8p rearrangement to the cells where we eliminated the 8p rearrangement, we saw 16 genes that were significantly upregulated in the duplicated region, 5 genes that were significantly downregulated that were expressed at very low levels in the deleted region, and then 340 genes on other chromosomes that were either upregulated or downregulated. So we saw almost 20 times more genes off of chromosome 8 that were affected by the inversion, duplication, deletion on 8p. And I think this underscores that 8p syndrome is it has significant impacts throughout the genome. And if you focus exclusively on the genes that are on chromosome 8p, you may be missing part of the causes of this syndrome because so many of the genes impacted by this syndrome are actually on other chromosomes. So I think these are two significant things that we found from this analysis. We went on to look at what sort of biological processes are affected by the inversion, duplication, deletion on 8p. We found a number of really fundamental biological processes that were downregulated in the cells affected by 8p syndrome. Things like translation, that is the production of proteins, were downregulated. DNA methylation was significantly downregulated, as well as a number of biological processes that are associated with neural development and embryogenesis. And we could verify the upregulation and downregulation of some of these genes using individual assays as well. The next thing that we wanted to do using these syndromes, using these cells, is we wanted to look at the effects of inversion, duplication, deletion 8p and compare them to cells from another chromosome disorder that's a lot more is known about and that's more commonly studied, which is Down syndrome. So we took our cells that have an inversion, duplication, deletion of 8p. We analyzed gene expression in them using RNA sequencing. And then we also used a published data set of gene expression in induced pluripotent stem cells with trisomy 21. And then we just compared the gene expression changes that were caused by inversion, duplication, deletion of 8p and the gene expression changes that were caused by trisomy 21. And we were really surprised to see that there were a number of highly significant gene expression similarities between these two syndromes, between these two conditions. So we looked at the genes that were upregulated by inversion, duplication, deletion of 8p and genes that were upregulated by Down syndrome. and the genes that were downregulated by both, and we found highly significant overlap. That is, in induced pluripotent stem cells derived from an 8p hero, and in induced pluripotent stem cells derived from an individual with Down syndrome, we saw highly significant changes in gene expression that are common between the two. And there were a number of fundamental biological processes that were similarly perturbed in both individuals. Things like the production of ribosomes, which are necessary for producing protein, DNA replication, splicing, fatty acid transport. Now, we don't exactly know why these processes themselves are downregulated. We have a few hypotheses, but we haven't dove into that yet. But on a fundamental level, I think that this analysis suggests that many consequences of inversion duplication deletion 8p are shared with other aneuploidies. And I think that this underscores that there's things that have been learned studying other developmental aneuploidies like Down syndrome, Edwards syndrome, Patau syndrome, Turner syndrome that can impact how we think about 8p syndrome, and that can think about how what the causes and consequences of 8p syndrome are. While there is significant individuality and uniqueness in these different chromosome alterations, there are some fundamentally similar or shared biological processes. And I think we can learn some things about 8p syndrome by looking at these other aneuploidies like Down syndrome, as evidenced by the fact that cells derived from an individual with Down syndrome share all of these gene expression changes compared to cells donated by an 8p hero. So that's what we found when we looked at the gene expression consequences of inversion duplication deletion 8p. The final thing that I want to tell you about today is work that was largely done in Stefan Pinter's group at the University of Connecticut, where we provided them with the cells that we had generated, where we had eliminated the rearranged copy of inversion duplication deletion 8p, and where they differentiated these cells into brain cells, into neurons. So the cells that we're working with here are induced pluripotent stem cells, and these are a fantastic research tool because they can be differentiated in cell culture in the laboratory into all sorts of different cell types found in the body. You can differentiate these stem cells into blood cells, into muscle cells, and into different types of neurons as well. And so if we believe that some of these processes are impacted in 8p heroes, studying how these cells produce how these stem cells can be differentiated into these cell types in cell culture can allow us to study this process, potentially do drug screens to find compounds that might enhance this process. These can be a good tool for research and the discovery of therapies. So Stefan's group took these induced pluripotent stem cells with an inversion duplication deletion of 8p, or in which that rearrangement had been corrected and restored to euploidy, and they differentiated them into neurons. So these are microscopy images of the neural differentiation protocol that we completed. The turquoise blue staining in these pictures is DNA. These are cell nuclei, so this just allows you to see all of the cells. And then the turquoise pink staining is MAP2, which is a marker for neurons. So basically, the more pink you see in these pictures, the more neurons were produced. And what we found is that cells from the proband, that is cells with the rearranged copy of chromosome 8p, produced significantly fewer neurons compared to cells in which we had eliminated the rearranged copy of chromosome 8p and restored two normal copies of chromosome 8 in these cells. So this tells us that cells with an inversion duplication deletion in 8p are defective in producing neurons. Um, this gives us a cell culture system for studying the production of neurons in cells provided by 8p heroes. And it also provides us a system for potentially doing things like drug screens or genetic screens to see if we can enhance the production of neurons in these cells. And Stefan's group had a great idea here toward that goal. There's a small molecule cocktail has recently been discovered that's been found to enhance the production of neurons in stem cells in general. So this is a group of 4 drugs called CEPT. These drugs decrease stress signaling and they decrease apoptotic signaling. So they suppress cell death. And a group of researchers at NIH found that the addition of CEPT enhances the production of neurons from induced pluripotent stem cells in humans. Stefan's group added CEPT to the cells with the inversion duplication deletion of 8p, and he found that adding CEPT enhances the production of neurons in the cells with an inversion duplication deletion of 8p. And in fact, in the presence of CEPT, there's no significant difference in the production of neurons overall between the proband and the revertant. So here are 2 box plots showing this. In the absence of CEPT, this is basically the experiment that I showed you on the previous slide. Cells with the inversion duplication deletion on 8p produce significantly fewer neurons than cells in which the rearranged copy of 8p has been eliminated. The cells with a normal karyotype produce significantly more neurons. However, when we do this neural differentiation protocol, when we treat cells with CEPT, which decreases cell stress and decreases apoptotic signaling, we significantly increase the production of neurons from these cells with the rearranged copy of chromosome 8p, and now there's no significant difference between these 2 cell populations. And so I think this is proof of principle, first, that cell stress and apoptosis is contributing to the poor production of neurons from the cells with the inversion duplication deletion of 8p. And that it's possible to use these cells that we've created in culture as a platform to look for therapies that can potentially enhance neurogenesis in cells with a rearranged copy of chromosome 8p. So that's what we found in the lab so far, and I'll take a few slides to kind of summarize what we've done and what I've told you about. So first, we received cells donated by an 8p hero with a rearranged copy of chromosome 8p, and we were successfully able to generate derivatives of these cells where we had eliminated the rearranged copy of chromosome 8p and restored a normal euploid karyotype. And I think this gives us now a great tool for studying the causes and consequences of 8p syndrome. We've been involved in sharing these cells through the foundation to other investigators who want to use this as a research tool for studying different biological processes in this condition. And I think that this is a cell culture-based proof of principle with many, 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 many more steps required. But I think it's kind of one small step towards a potential therapeutic approach. Because if we can correct the aneuploidy in cell culture, I think that's the first step towards potentially correcting this rearrangement in an animal model or potentially in patients after many more steps are completed. Secondly, we use these cells as a tool to study the effects of 8p syndrome on gene expression. We were surprised to see that most gene expression changes in these cells were not on chromosome 8. 8p syndrome is really a genome-wide condition. There are cells on chromosomes, 1 through 7 and 9 through X that are also impacted, upregulated or downregulated in cells from these individuals. We found that this chromosome rearrangement affects several biological processes. And we also found that 8p syndrome shares many transcriptional similarities with a different developmental aneuploidy with Down syndrome. And so I think we can learn things about 8p syndrome by looking at these other developmental aneuploidies as well. Finally, we characterize the production of neurons from cells with the inversion duplication deletion on 8p. We found that 8p rearrangements by themselves significantly decrease the production of neurons from stem cells, which you can tell by the decrease in pink staining here, which is a marker of neurons. However, we also found that a small molecule cocktail that suppresses stress signaling in induced pluripotent stem cells was sufficient to significantly increase the production of neurons from these inversion duplication deletion 8p cells. And I think that this tells us that some of this stress or apoptotic signaling may be contributing to defective neurogenesis in these cells. And it's proof of principle that we can use these cells as a drug screening platform to look for potentially therapeutics that could be repurposed towards this goal of enhancing neural production in these cells. So thanks so much for your time today. Again, I want to reiterate that the experiments that I showed you were done by Sophia in my lab and then Stefan and Aaron at the University of Connecticut. We're incredibly grateful to Project 8p for supporting this research, to the 8p heroes who donated their cells to this study. If you're interested in learning more about this work, we have a preprint up on bioRxiv right now where we go into more detail, including all of the negative data about the chromosome engineering approaches that we attempted that didn't work before we found the ones that did. And also, if you're interested in chatting further, feel free to reach out to me. You can email me, my last name, sheltzer@stanford.edu. And I'd be happy to meet with any members of the community, any affected families more, to talk about this in more detail. So thank you so much again. Happy to answer any questions that you have.
Thank you so much, Dr. Sheltzer. That was a phenomenal presentation. We're so grateful. At this time, if anybody would like to unmute and ask a question directly, or please feel free to put it in the chat as well.
I have a question. Hi. So there are different rearrangement types that are in. So this one was an inverted and with the deletion. So is there plans to do any other of the types? Because I would think, it'd be interesting to see how they're the same or different, to see whether or not you can identify something that's common among the rearrangements.
Yeah, I think that's a fantastic question. And that's one of the things that we're very excited about taking a chromosome engineering approach to look at 8p syndrome. So this plot shows, the diversity of rearrangements that affect chromosome 8p. You can have deletions of different sizes, duplications of different sizes. Our hope is that by targeting chromosome 8 itself, that this pipeline that we generated for restoring a new normal euploid karyotype, that we can now apply this to cells with different rearrangements on chromosome 8p, and basically the same thing that we did with the cells from this 8p hero should ideally work for any other type of 8p rearrangement. So our hope is that this will be a kind of generally applicable strategy for looking at other rearrangements affecting chromosome 8p. And we will be moving forward with this. We will be receiving additional cells from Project 8p, and we're going to be continuing this work to look at least cells from one additional 8p hero and potentially from more 8p heroes as well. So yes, we think that it will work in other 8p rearrangements, and we will be moving forward with that work in the next few months.
Do you feel that you have to go through the whole process with each cell line, or can you skip a few, like look at the underexpression or overexpression of things that you think are highly candidates? Or do you really have to go through the whole thing because of, background issues and things like
Yeah.
that?
I think that's another really excellent question. One thing that we found from this is that so when we just looked at cells from an 8p hero and cells provided from their parents, we found 1,400 differentially expressed genes. We found hundreds of genes that were upregulated and downregulated. And these were not caused by the 8p rearrangement. These are just caused by the differences between a parent and child. And so we think that when you do these sorts of comparisons across genetically diverse individuals, you get all sorts of changes that are caused by the SNPs, the random point mutations between individuals, and that aren't specifically caused by 8p. So we think that to have the cleanest results, the most rigorous results, we do want to go through the chromosome engineering process to create this euploid revertant population. At the same time, I think that using what we learned from this successful test case, ideally we'll be able to do it a lot more quickly for the second, third, fourth, and fifth times that we're doing it. I think Faye has a question.
I do. Thank you so much. I mean, this is absolutely incredible work. So, as a parent and a scientist, I respect it. So amazing. Thank you. I think my first question is actually just a clarification. Did you say that all the chromosomes in your corrected cell line were the same? So like, it was the same version of chromosome, like 1 like duplicated and chromosome 2 duplicated, as well as the same version of chromosome 8 duplicated. Did I understand that correctly?
So when I said that the chromosomes 1, 2, 3 were the same,
Okay.
what I meant is that they're the same as those chromosomes in the 8p hero. So the
okay, so
chromosomes
still
themselves
different.
are still heterozygous. You still have the maternal and paternal copy. They're
Okay.
identical to those provided by the 8p hero.
Okay, great. And so then with the actual chromosome 8, what exactly did you have? Did you have 2 copies of the same normal chromosome 8 of the proband?
Yep. So terrific question. So we have an isodisomy of chromosome 8 now.
Okay.
So
Is that what it's called?
yes. So the 8p hero inherited a normal copy of chromosome 8 from one parent and a rearranged copy of chromosome 8 from the other parent. The cells ended up with a duplication of the normal copy of chromosome 8 from one parent. So they have an isodisomy, 2 copies, diso of the same as iso.
Actually, I didn't even know there was a term for this. This is excellent. And then my second question relates to the this sort of neuronal differentiation you did. So, or Stephen Pinter's lab did rather. And so I think I'm just curious. I mean, that's an incredible result where, you showed the sort of normal neuronal or nearly normal neuronal marker where you differentiated the revertant cell. And you were talking about sort of lack of cell stress, and would you say lack of apoptosis? I think I'm kind of curious about the cell number. What did the plates actually look like, with the cells in them? Were there more cells when you were sort of growing out the proband, sort of unreverted? Do you kind of understand what I'm asking? It's a
Yep.
bit word salady, sorry.
No, I totally get it.
Yeah.
So there were fewer cells overall in the cells with a rearranged copy of chromosome 8.
Okay,
So
so they just weren't surviving, I see.
yes. However, so there were moderately fewer cells overall, but the difference in neuron production was significantly greater than just the overall difference in cell number. So we think that the process of neural development is a very stressful process for these cells. And so if you have kind of basal level of stress from the presence of this rearranged chromosome, that the neural development may enhance that stress and lead to apoptosis or cell death. And suppressing that apoptotic signaling can help the cells survive this stress and get to the mature neuron state.
Well, I think I'd be really keen to see experiments as well, where you just sort of like reduce the amount of, I'm sure there's drugs you could have where you just reduce the amount of translation. Would that also reduce the stress, because presumably it's the extra noise coming from the duplication that is adding that to that stress.
Yeah, I think that's a great experiment. One of the drugs in this CEPT cocktail is a translation modulator.
Okay.
And so it could be through the modulation of protein translation. That could be an important component of why the CEPT cocktail was able to rescue neural production.
Makes total sense. Thank you so much. Really brilliant work. Congrats, everyone. Thank you.
If I could ask one, but please feel free. I am lucky enough to talk to Dr. Schultzer frequently, so please feel free to jump in if you have other questions. But I'd love if you could give us a little bit more background about where the CUPT cocktail has been used, and maybe a little bit about, it's not necessarily a fair question, but there's four things in it. So one could think about disentangling the benefits from each one of these things, or whether or not there's some synergy that emerges that is greater than the sum of its parts, and kind of what we think about, kind of teasing that apart.
Yeah. So uh, human stem cells have been isolated and worked with for about 25 years. And basically, as soon as stem cells were first successfully isolated and turned into neurons, people have thought about making neurons in cell culture as a potential therapy. So, for instance, if someone had a stroke, people have thought, maybe you could take their cells, produce some neurons in cell culture, and then inject them into the brain as a stroke treatment. And all of that sort of, research is ongoing. Towards that goal of being able to make neurons really efficiently in cell culture, researchers have looked at strategies to enhance the production of neurons from stem cells. And there was a group at the NIH who published this paper 3 or 4 years ago here in Nature Methods, where they found this polypharmacology platform, basically CEPT, which protected cells and allowed them to form neurons better in cell culture. So these researchers were not thinking about aneuploidy, they were not thinking about 8p syndrome. They're thinking, how can we make neurons most efficiently in cell culture, towards making a treatment for stroke or making a treatment for some other neurodegeneration condition. Um, the components of this, so polypharmacology means, has a lot of effects, basically. These are not, highly specific drugs that you would take to inhibit a specific biological process. These are drugs that are known to be somewhat promiscuous and to impact a lot of different cellular processes. So these are maybe not the best from a therapeutic perspective in terms of giving to a person because these are not precisely engineered drugs. These are things that impact, dozens, if not hundreds of biological processes, and they could have all sorts of off-target effects or potential toxicities in other organs or things like that. So I think that's a downside, but it does point towards stress and apoptotic signaling as a key, gateway towards the production of neurons in cells from affected by alterations on 8p. And so potentially in follow-up drug screening work, looking at pharmaceutical grade or therapeutic grade compounds that are known to target cellular stress and apoptotic signaling, that might be a great approach to move this towards a therapy. Though we are, we are not there yet just with this cell culture experiment.
Ah, well said. Oh, Internet, I see you have your hand raised.
I just had a quick question and maybe I missed it, but what was the 4 small molecules that were in those cocktails?
I would need to, let me see, I have that written down somewhere. Let me just find it.
And like a quick follow-up, are we essentially
Mhm.
saying that you added these to culture and it allowed like it allowed you to make a stem cell line, or it allowed stem cells to be turned into neurons more like with a higher rate than if you didn't use it?
So the small molecules are chromin-1, emricasin, polyamines, and trans-ISRIB. Trans-ISRIB is an inhibitor of the integrated stress signaling pathway. This is the translation modulator that I had previously mentioned. And then chromin-1, emraskin, and polyamines have all sorts of different biological effects that are thought to impact apoptosis and stress signaling. We added CEPT during the neural differentiation protocol, and they enhanced neural production in both the affected cells and the cells with where we had eliminated the rearranged copy of chromosome 8p. So they had an effect in both conditions. And at the end of the process, there was just no significant difference between them because they had elevated the cells with the rearranged copy of 8p to the wild type levels.
Okay.
Oh, attacking on mute again. That brings us to the top of the hour. Thank you all so much for your time and attention. And we really appreciate it. We'll be sending out more details for our next research roundtable and hope you join us there as well. Thank you all.
Thank you so much. Feel free to reach out to me directly if you have any other questions. And again, thank you, Bina, Lauren, for the support of the foundation. Really looking forward to continuing our work together.
Our pleasure. Thank you, Jason.
For sure. Thank you.